bit of a study guide there with sample questions, and that you can use that to prep for the test. And before the test comes, you'll have an on Q&A for the test. I'm not gonna let you flounder after that. So don't worry about it. Yeah.
What about a book? What should we be reading a chapter a week? And tell me what should we be doing with it?
What I would do is follow through the syllabus. In the syllabus you have, and then also the calendar for the timeline of what we're talking about. So in the first test, everything introduction, microscopy, and the section on prokaryotic structure and function. That will be all on the first test. The second test you'll see is much more molecular in nature. That's the section on the catalyst. So you want to read along the chapters in the book, do the problem sets in the book. Those will be very helpful. And also listen to what we're saying. Oftentimes I will, or sometimes I will mention stuff that's not in the book. The other way is probably also true. That sometimes the book mentions stuff that I'm not going to talk about. So you will also find maybe that the book explains stuff in a different way that I do. You may find that easier, better to understand. Another person may find what I have to say more easy to understand rather than the book. So I think it's a complementary approach. If you read the book alongside your lecture notes, that definitely will clear you. When it comes to lab techniques test, that's a different issue, where we'll just practice lab techniques, but in terms of the lecture, we have clearly defined modules, and the first module is for second module is all of this belong to these individual modules that should be good. And again, we'll last class before the test we'll do last time ask the questions to the last time too. That's what I'm there for too. You do your experiment, I have nothing to do. Keep me occupied. By all means, please do that. Don't shy away from it. Hey, you should know that's perfectly acceptable. Any more questions in terms of organization? Alright, so let's get going on the program. Or not. So if we just had to compare prokaryotic and eukaryotic cells, this slide really has it all. The common feature of prokaryotes are they don't have a nucleus. In fact, they don't have any membrane-bound organelles. The only membrane that you find in a prokaryote is the plasma membrane, the cell membrane, which means they have no organelles, nothing on the inside. Whereas the eukaryotic cell that does have membrane-bound organelles, and those include the nucleus, the mitochondrium, your plant cells, the chloroplast, and the plasmatic reticulum, the Golgi apparatus, blah 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 blah. So when we go over eukaryotes, I'll introduce all this eukaryotes, those eukaryotic organelles separately. But that is one of the biggest distinctions. No membrane-bound organelles in prokaryotes. Since they don't have a nucleus, they also do not have chromosomes, or rather they don't have linear chromosomes. Prokaryotes have exactly one chromosome, and that is a circular one. Whereas eukaryotes usually have linear chromosomes and have multiple versions of them. Humans have, for example, 23 chromosomes. Eukaryotes, with the exception of plant cells, also normally don't really have a cell wall. If eukaryotic cells have cell walls, they can polysaccharide. Plant cells is cellulose, for example, the plant cell wall. Prokaryotic cells invariably have cell walls, mostly made of a substance called glycan, we'll talk about that in a second. Whereas archaebacteria have a slightly modified version of that acetoglycan cell wall, which is made from pseudomurine. Again, I'll show you how that looks like. And the way they reproduce is also very different. The process of eukaryotic cell division is called mitosis. It's a very complicated, drawn-out process, whereas a prokaryotic cell simply divides by addition, just divides the two. It's very simple. What's not on this slide here is that prokaryotes can also have sex. Sometimes you have prokaryotic cells that have express certain sex factors, and then they extend a little outgrowth in the cell membrane to another cell, and they exchange a genetic material through that, which is kind of cool if you actually understand kind of a bizarre arrangement. What prokaryotes also have, they have DNA in addition to their chromosomal DNA. They have extra chromosomal DNA, and this is called a plasmid. Many prokaryotic cells can, through this process of serial sex, can exchange plasmid DNA. And that is a big contributing factor in spreading antibiotic resistance, for example, through bacterial cell populations. Because they can simply exchange those plasmids, which would carry those antibiotic resistances, would simply just exchange that. So, part of your job in lab, today and for the next couple of weeks, will be to be able to differentiate bacterial or prokaryotes simply by size and shape. So for that, you need to know what kind of sizes and shapes bacteria come in. Usually, they're not much longer than 2 micrometers in diameter, or maybe up to 8 micrometers in length. If you remember from last time, how much a micrometer is, 10 to the minus 6 meters. A meter is about really long, so you can kind of see how small they are. We've seen millennia and the other thing was that someone help me. Thank you. Paramecium last time. You kind of have an idea how big these sizes are. So when we go to the lab later on today, you'll see bacteria, and they will be much smaller than the eukaryotic cells we had last time. And the size and shapes of these guys are fairly simple. They usually come in just 3 major shapes and forms. Either the cell shape, which is more like a rod shape, like my laser pointer here, the simple rod. They come in coccus variety, which is just a big ball. And they can come in spiral forms, in the Vibrio spirillum or spirocate variety. These are the major forms of bacteria. And usually bacterial populations of a particular genus simply accept that shape and don't change. Very rarely, or kind of not so common, also star-shaped and rectangular forms. So the subgroup of the spirobacteria, for example, would look something like that. So this would be Vibrio, which is a little bit of a bent rod kind of thing. Okay? This would be Aspergillium bacteria, which is really very oddly shaped. It's like a screw. That's how it kind of looks like. And then the spirochete is a little bit smaller, but in essence also just a big wiggly line. So these kind of things, just based on that, you can classify your prokaryotes based on that. And then very quick one, star-shaped bacteria. I don't even know what this is, but they're really cool. I've never seen that in real life. Rectangular bacteria. So these are more the rarer kind. What will work in the lab are mostly the cocci and the bacillus type bacteria. Okay. So this is the basic shape. Either rods or round balls, or maybe a little bit of a twisted rod. They can also aggregate. So when they aggregate into pairs, have basically two of these guys living together and forming that organism. If you have clusters, you have, we call it a staff. So a staff of bacteria would be a staphyl. That would be the prefix for that. So staphylococcus kind of bacteria would be something that takes up a round shape that likes to cluster. If bacteria like to form chains, we talk of a strepto. You prefix that with a strepto. So you can have streptococci, they form little strands of beads, or streptobacilli, which form strands of rods. Yeah.
How should the shape and arrangement of these bacteria determine the medium in which they thrive?
Their shape does not determine the medium. Their medium may determine their shape, but you have such a variety of bacteria, millions of bacteria right as we speak, that there's really no clear correlation between what kind of growth medium they use and what kind of shape they take on. But let me say that under certain conditions, bacteria tend to cluster. That is a defense mechanism in a sense. You may have heard of a biofilm. That is a very dense population of bacteria that clusters together and forms a thick layer coating something. So it basically becomes a very slimy surface. So that's a biofilm, and it helps bacteria survive harsh environmental conditions. So that would be the only clear-cut example where we can say, yeah, this particular environment forces kind of arrangement of bacteria. Okay. And then, of course, if they can arrange themselves in pairs, they can also arrange themselves in fours or in groups of 8. Then we call that a tetrad or circinate. Basically, this is just when cells divide by fission, they double themselves in size. Where simple cockles could, just by the pure act of doing addition, turn into a diplococcus. If this diplococcus again undergoes fission, then you have the tetracoccus, because now you have 4 of those cocci living together. But that goes into the description of the bacteria that you're seeing, especially today when you're doing a couple of stains on a whole variety of bacterial strains. That goes into your description, how they look like essentially. So here are some examples. Diplococci, of course, 2 of these cocci, see the XX and 2. Streptococcus strain could form long elongated beads on a string kind of thing. It can form a tetrad, it can form a circinate, basically 4 or 8 clusters, and then the staphylococcus strain would really just grow in big clusters of tens, hundreds of cells. Same can be done with bacilli. Single bacillus is a single rod, as you already see here. These are diplobacillus here, these are diplobacilli. And you can have also coccobacillus, which is not quite as rod shaped, and it looks a little bit like a cockroach because it's a little bit rounder. But again, the same principle applies. Multiple of the bacilli work together and they're shaping their changing their shape. Close up of a group of diplobacilli, and then the whole streptobacillus, the rod-like structures that form a big chain where they now arrange themselves in a big long elongated So that is highly dependent on also the genetic makeup of the bacteria. If they really like to cluster together, then they're definitely in culture you will see this clustering happen. And that's why staphylococcus strain is called staphylococcus, because they just cluster together in their growth medium. Whereas the streptococcus really just will grow in these individual pieces. Okay, this is just morphology, description of how these things look like. So for example, if you were to look at a particular bacillus, that will have a bacillus shape, which is the rod-like shape. This particular bacillus is called a Bacillus anthracis, which is the anthrax bacterium, which, if you remember, was used a couple of years ago to do some, I guess, scare tactics, terror attacks kind of thing. These would be those bacteria that cause lung disease and eventually will kill you. Anthrax usually is lethal for cattle, but they can also cross-react with humans and cause a fairly lethal lung disease now. So this is how the anthrax bacterium is. Really nothing special, it's just a bacillus. Okay. So what we'll do today, and which is where I'll take a little detour right now, is we'll perform simple staining of cells. The reason for perform staining is that, as you've seen last time, these guys were very difficult to see. The eugenia was fairly straightforward to see because it was green, right? It has the chloroplast inside, which are green, so the visualization was easier. But the paramecium, that was fairly confusing. And now consider bacteria which are quite a bit smaller than these eukaryotic cells that we looked at last time. And they're just as translucent, if not more so, because they don't have any organelles on the inside that can additionally refract the light, which makes it already very difficult to see them if you shine a lot of light through it. If you remember, or if you recall from last time, when you had your lights really high up and I walked by and said, no, turn it down, turn it down, turn it down, and all of a sudden saw more, that will be exacerbated now with this vision. So even if you turn down your light a lot on the microscope, you may still have a hard time seeing. And this is where stains come in. So with stains, you just put colored dye onto the cell, and all of a sudden you can stand out from the background. The only problem with the stain is that it usually kills your cell. So if you want to see what it does, using a stain is very tricky, or if not outright impossible. But if you just want to look like it, see what it looks like, or if you work in a hospital and you get a culture from a patient, you want to know what bacterium he's infected with, then you just simply do a simple stain, and you can figure that out. You're not interested in how to do it. So you can also make the staining very specific for particular cellular substructures. You can use stains for DNA, for example, that would stain a nucleus, or use the chromosome in a bacterium. You can, and we'll do that next time, have particular stains for bacterial cell wall, and so on. So depending on what kind of stain you're using, you can also highlight certain substructures in that bacterium that may give you an additional clue what kind of bacterium that is that you're looking at, and what kind of countermeasures you may want to take to get rid of that. In order to stain something, first you have to fix your bacteria on your covers, not on your covers, on your slide. The way we do that, and I'll show you later in lab how this is done, take a small scraping of bacterial material, smear it out on your slide, and then pass it through a flame to fix the bacteria onto that slide. So basically, you can mobilize your cells onto that slide so that when you add your stain, you don't wash it off the slide. Basic stains can be, or simple stains can be the basic type, where the chromophore, where the color dye is a cation, or an acidic dye where the chromophore is an anion. That's why I was talking about in the first class that we have strong acids and bases in class. Most of your stains have acidic or basic, which also means they only stain certain substructures. If you have a basic dye, would that stain, or would that attach to a structure in the cell that is mostly cationic in nature? So cation is something that has a positive charge, right? dye on that, that would not stick, because the basic dye itself is positively charged, and if you have a structure in the cell that is positively charged, it would reject the cell, so you couldn't use that. But you could use an acidic dye on a structure like that, because an acidic dye is negatively charged, that would stick very well to that positively charged structure in the cell. So depending on what you want to stain, you also may want to carefully select your stain if you want to work with it. Not every stain may be useful. If you stain in the background instead of the cell, it's called a negative stain. You can do that to highlight the bug against the background of the thing. And you can do that with stains that will not stain any cellular structures, but only your slide. Again, judicious choosing of your dye will do that. Glass is usually, you can buy specially treated cover slips and slides that may come with a positive charge already on top. So if you use your stain, the appropriate stain for that, the stain will stick exactly only to your slide, but not to your cell. This is how you could potentially use a negative stain to highlight. What we'll do today is a simple stain. We just use a simple one dye stain, dump it on your cells, everything in the cell will be stained, and then you can just simply see how the cells look like in their structure. You can also use something that's called a mordant. That is something that increases affinity of your cells for that stain. Basically, increases contrast, makes the stain stronger and stronger. There will not use a mordant. Okay. Just to give you an idea about the procedure. So here, I was mentioning, if you have unstained specimen, it's really, really hard to see. Your light goes right through, but if you stain it, it actually makes life so much easier to detect it. And as you're staining it, there's a couple of different ways of staining. We have simple stains, differential stains, and special stains. We will do all of those in lab. Today we'll just use simple stain, and the easiest simple stain is a simple dye, crystal violet. Basically, you have your bacterial smear on your slide, you add your crystal violet to it, let it stain, wash the dye off, and lo and behold, your cells are stained. That's essentially what we'll do today. I think we'll do a crystal violet stain on most of those, which has, as the name said, a slightly violet color. You add it to your cells, wash it off, and so on. Not a few other simple stains, a separate stain, which is a bright pink stain that you can add to your cell again. You add dye, you wash it off. You get the idea. And finally, methylene blue, which is a blue dye, which is used in a variety of biological applications. It's very dark blue. You add it on, wash it off, and all of a sudden your cells look blue. We don't have to talk about differential stains now. We'll do that next time, especially when the lab comes up for that. But that is the basic procedure, and that's the basic procedure that you'll do today. You prepare a smear, you add the dye to it, you wash it off, and look at it under the microscope. Which brings us to the next part. What are we looking at? What are the structures in the bacteria that make the bacterium prokaryote to prokaryote? So in general, this is how prokaryote looks like. In the center of the cell, you have your nucleoid, which is not a real nucleus, but it's kind of a little bit of an area where the single circular chromosome is contained. In the cytosol, the cytoplasm of the cell, you have lots of these ribosomes where proteins are made. All of that is surrounded by a plasma membrane. This plasma membrane has flagellae attached, maybe some cilia or fimbria attached as well. Just outside of that plasma membrane, you find the cell wall. And in many bacteria, you have even a further layer outside the cell wall, which is kind of a sugar coating or a capsule, also sometimes called. So in in terms of structure, they're really exceedingly simple. There's really not much going on here. Oh, and there's also this pilus here. Cells sometimes use these pilae for the exchange of that plasmid DNA that I was talking about. Plasmid is shown here, extra chromosomal piece of DNA that is not in that nucleoid in the cell. And we'll talk about those also as we discuss prokaryotes. So on the outside of that cell wall, that capsule thing is called the glycocalyx. The glycocalyx is called glycocalyx because it's made from sugars. It is very viscous, it's a little bit like jello, and it protects the cell wall from the environment. You can differentiate two types of this glycocalyx thing. One is called a capsule, the other one is simply a slime layer. But in essence, it's very similar. A capsule is a relatively firmly attached version of these polysaccharides, whereas a slime layer is a little bit unorganized. And what this does is protects the cell from the host immune response, essentially, from the environment. Because having a capsule around a bacterium really prevents an immune cell from recognizing that cell as a foreign antigen. And when we talk about in the last section of class about the immune response, we'll come back to that. So immune cells basically cannot recognize it as foreign, cannot eat it, cannot kill it, and basically this is a way for the bacterium, for the prokaryote, to evade the immune response of the host. This also contributes to the formation of these biofilms that we're just talking about. Under certain conditions, bacteria come together and form these biofilms. And again, that's the glycocalyx of the bacteria, all acting together, being secreted, making a big slimy layer around the cell. And this is how it looks like in a microscope. You see clearly a very highly refractive layer just around your bacteria, and that is the capsule. Very, in this case, a highly organized, relatively solid form of jello around the cell. In terms of what it's made from, we talked about agarose gels and all that. I don't have, these capsules can be highly variable in the sugars that they use. I'm just throwing up a picture of agar and agarose. If you've ever run a DNA gel, that's what you're using, or the plates that you're using that you touch with your fingers in the first class. Here, the gelling agent is agarose. And essentially, that is the same consistency that the capsule around the bacteria has. A little bit softer maybe a little bit firmer depending on what kind of strain of bacteria you have. But that's exactly what it would feel like if you were to touch it. Below that glycocalyx is the bacterial cell wall. The bacterial cell wall serves one important function. It keeps the cell from breaking down, especially under situations of osmotic stress. And we'll talk about osmosis again as part of a discussion of bacteria. So during osmotic stress, cells may rupture. If you have a bacterial cell wall there, cells will be protected from rupture. So it's very advantageous for a cell to have a cell wall like that. A component of the cell wall is a molecule called peptidoglycan, especially in bacteria. This peptidoglycan is one major contributor to disease-causing bacteria. The immune system likes to, or specifically recognizes peptidoglycan. And again, that's the reason why some bacteria have a capsule around to protect those peptidoglycans from the immune cell. And it contributes massively to virulence and other things. Now, how does it look like? If you look at a cell, you can again clearly see the calyx, the capsule around it, it's a little fuzzy. And here, this thin line, this very dense line here, that is your cell wall. What's the cell wall made from? What's peptidoglycan? Again, it's a sugar. kind of the theme in nature. Sugars are used when you want to build stuff that is very, very strong. Same here. The sugar that makes the bacterial cell wall is called peptidoglycan, which is a polymer of two individual sugars, NAM and NAD, N-acetylglucosamine and N-acetylmuramic acid. These alternating NAD-NAM sugars are linked together by amino acids, by polypeptides. And therefore you're creating a three-dimensional network, a three-dimensional lattice that really encapsulates the rest of the protein. Here are your sugars in action. This is N-acetylglucosamine. This is N-acetylmuramic acid. If you've ever seen a glucose molecule, that looks very much like a glucose molecule. So these are basically just substitutions with some acetic acid here on the regular glucose molecule. So the same thing that makes your coffee sweet makes the bacterial cell wall. So that's kind of cool. And then, especially here, the cross-linking between individual strands of sugar. So this is a polymer where NAD and NAM alternate. These polymers are 10,000 individual, 10 to 50,000 individual sugars are linked up to form a single peptidoglycan. And then individual peptidoglycan molecules are cross-linked to each other through a pentapeptide. These are 5 amino acids in a row, and this is one of the very few examples in nature where you actually have a 3 amino acids, all amino acids, and usually in the L form. This is one of the rare instances where you have actually the D form, the mirror image of that. So everything in nature is chiral, which means it has mirror images, and one of the mysteries that scientists cannot quite explain is why a particular mirror image is prevalent in nature. Here, you actually see both E-alanine and L-alanine, both mirror images of amino acid alanine are present. So that is kind of interesting. So having these amino acids, this pentapeptide, this 5 amino acid peptide, hooked up to muramic acid, to that muramic acid group here, leads to cross-linking of these individual peptidoglycan molecules. The way this looks like is shown here. So here's your NAD-NAM polymer, alternating polymer. Here you have your penta amino acids on the muramic acid, and now you have cross-linking through a polyalanine chain here in the middle. So these peptide chains that are hooked up to the sugars are in turn hooked up to some peptides, and that creates the cross-linking of these individual sugar chains. Okay? It's a pentaglycine bridge, I'm sorry. I should say that. So here, 5 of these amino acid glycines are used to bridge the gap between individual muramic acid linked peptides, and thus creating a cross-link section, and that builds the bacterial cell wall. You can have many of these cross-links, then you have a very thick cell wall, or you just have a very few, then you have a very thin cell wall. And when we talk about gram-positive and gram-negative here, that's exactly what we're talking about, the thickness of these cross-links, how many of these individual peptidoglycan units are linked up with each other. Another way of showing that is right here is essentially the same thing. You have your NAD-NAM polymers hooked up to your D-alanine containing side chain, and then you have these polyglycine cross-links that connect these individual peptides to form the eventual cross-link. If you extend that in all 3 dimensions, you can see you can build a really strong lattice around the bacterium. Makes sense? So in bacteria usually come in 2 flavors, gram-positive and gram-negative flavors, and we do gram-staining lab section. And that is simply a measurement of how thick the bacterial cell is. A gram stain will stain especially that peptidoglycan. So if you have a lot of peptidoglycan on the gram staining, that means your bacterial cell wall has many of these polymers, it's very thick, has lots of these magnetic cross links with each other. If you have a gram-negative bacteria, that means you have a very thin bacterial cell wall. The dye that is taken up by the peptidoglycan has very few peptidoglycans to bind to, and therefore it appears not stained. But even gram-negative bacteria will have a modicum of bacterial cell wall, just a very thin one, not as highly cross-linked, not so many individual monomers of that. Gram-positive will have a very thick bacterial cell wall, will have many of these cross-linking units, and you see very thick. Therefore, we get a lot of that gram stain. The basis of the gram staining is the retention of a particular dye by the bacterial cell. Some of them will retain a lot because there's a lot of cell wall, and therefore we call those gram positive. Okay. Another subgroup here will have a very thin bacterial cell wall, will not retain so much of their dye, and therefore we call them gram negative. The other thing about gram-negative bacteria, again, we'll talk about that later, is instead of that thick bacterial cell wall, you have an additional outer membrane. And that's why they don't need such a thick bacterial cell wall, because they have an additional membrane. Okay, so here's the difference between the gram-positive and the gram-negative cells. Gram-positive cells have very thick peptidoglycans, whereas gram-negative have very thin peptidoglycans. The gram-negative cells, in addition, have an outer membrane around that whole structure that we just discussed. Here. This is your bacterial cell wall. It has an additional membrane that sits even outside of that. Which also creates a space between the outer membrane and the inner membrane, which is called the periplasmic space, which just means the in-between the membrane space. And the gram-positive cells don't have any of that. They have also teichoic acid in their cell wall, but these are just different ways of crossing it. So teichoic acid, yes.
So for gram-negative cell walls, does that outer membrane be glycocalyx layer?
No, a membrane is not a glycocalyx, right? A membrane is made of lipids, whereas the glycocalyx is made of sugar. So in the case of a gram-negative cell, a glycocalyx would sit on the cell wall. So gram-negative don't usually have a capsule like that. So the other thing that gram-positive cells therefore has is also molecules of teichoic acids, which are lipoteichoic acid links cell walls to plasma membrane. Okay. What that means is that teichoic acid are cross-linking agents that really hold the bacterial cell wall in place. They can cross-link the bacterial cell wall with the plasma membrane by being anchored on one side in the plasma membrane through this lipo group here. Lipo just means fat, and through the acidic group, it can interact with the bacterial cell wall, therefore creating a cross-linker that holds the bacterial cell wall in direct contact with plasma membrane and holds it in place so it doesn't fall off accidentally or by mechanical disruption. So both the bacterial cell wall and the teichoic acid are the substances that you use it recognizes a bacterium as form, as not part of the biology. Because the three-dimensional structures of these sugars are so distinct that it easily recognizes that something is not of your own. And again, once we talk about using that debate a little bit more sense. So then the three-dimensional structure of such an outer layer of bacterium or prokaryote would look something like that. You would have your plasma membrane with your phospholipid bilayer here. You have proteins embedded into that plasma membrane. On the outside of that plasma membrane, now you have your peptidoglycan in a three-dimensional, highly cross-linked fashion. And that is held together, held in place in relationship to the plasma membrane through this teichoic acid cross-linkers that hook up to the plasma membrane that are connected, directly inserted into the plasma membrane. And then through the acid group also interact with the bacterial cell wall, providing a three-dimensional structure, making sure that it stays in place and on the outside of the cell. The gram-negative cell wall will look slightly different. We talked about how they have an additional outer membrane, which is called, which creates a periplasmatic space or a space between membrane. And the outer membrane is again, it's just a membrane. Polysaccharide, lactobacillus, phospholipid, just like any other membrane. We'll talk about membrane, how they're made later in this class. So a gram-negative cell wall has an advantage over a gram-positive cell wall in a similar way that a capsule provides yourself an advantage. It's an additional layer of protection. In contrast to peptidoglycan cell wall, which has these structures, very rigid three-dimensional structure and such, which provides recognition sites for to attack it. The plasma membrane itself is a relatively smooth affair, just like a capsule would. Therefore, that outer membrane again not to be eaten by cells in the environment, phagocytes, eating cells, T cells that would kill it, and so on. So again, this is a way for a bacterium to evade an immune response. So if you ever work in the hospital, one of the most important stains that you do in the hospital is the gram stain, because you want to know, do they have gram-negative cells in that patient, or do you have gram-positive cells? A gram-negative cell is very hard to get rid of because of that additional protective function of that outer membrane. Gram-negative cells also have an additional problem, and that's called lipopolysaccharide, or LPS. LPS contains lipopolysaccharide is exactly what it says. A polysaccharide that has fatty acids or fat-soluble things attached to it. So the structure of lipopolysaccharide is something like that. Again, you recognize your sugars here. They're very similar to glucose. These are your sugars, and on top of that, you have this very complex, don't worry about it, kind of sugar tree of hundreds of different sugars. That's the polysaccharide portion of that. The lipopolysaccharide portion, however, is the problematic one. So here you have a couple of fatty acids. So this is a very lipid-like structure. So that will insert into the plasma membrane of the bacterium, and the sugar groups here will seek out into the environment. However, that sugar group can be cleaved off by enzymes, and then you end up with this base region. This is also called lipid A or endotoxin. If you've ever worked in a hospital for a doctor, or eventually will work in a hospital for a doctor, that's one of the most feared words that you can ever tell any medical professional. I've detected endotoxin. Endotoxin will lead to a massive immune response from the immune system to the point that this will kill you. This is also called septic shock. You may have heard of that. When your intestines get punctured, that means all the bacteria that are currently living in the intestines go directly into your bloodstream. That is what sepsis is. Your intestines get punctured. Most of those are gram-negative and have LPS on their surface. Now, endotoxin will get your immune system overdrive to the point where it overreacts and actually kills it. Endotoxin is one of the most toxic substances that we know of. So all surgical supplies, all medical supplies and whatnot have to be absolutely endotoxin free. And you will probably not enjoy that. The only way we can test for endotoxin is by doing that. What's that? That's the limitless amoebicide lysate endotoxin test. For some reason, we don't have any good analytic capabilities to detect endotoxin system. However, the horseshoe crab, this little creature here, has in its blood, they don't really have blood like we have, where we have T cells and B cells and whatnot. They have something in their blood that's called an amoebocyte. If you remember paramecium from last time, that's an amoeba. So their blood cells are very similar to that. They kind of walk around in the blood and do stuff there. And these amoebocytes are highly sensitive to endotoxin. When you expose them to endotoxin, they immediately coagulate and you get a color reaction. That is the most sensitive test for endotoxin that we currently have. So how do we get these amoebocytes from the mollusk? The horseshoe crab. We basically catch horseshoe crabs from the ocean, put them in little racks in the lab, and bleed them. That blood is blue, by the way. Kind of cool. So you take their blood off and then put them back into the ocean. The companies say 95% of horseshoe crabs survive. That may or may not be exaggerated. Just to give you a perspective, the horseshoe crab has been around on the planet for 500 million years unchanged. So we're using that to test endotoxin, which is bizarre. So this is LPS, lipopolysaccharide. Again, this endotoxin portion here, that is really the problem of gram-negative cells, because they have that in their membrane and it's highly toxic. So the gram-negative cell wall looks something like that. Again, you have the inner membrane, you have the periplasmic space between the two membranes. You have a very thin composite lichen layer. You barely can have the gram cell. You have your outer membrane, and into that outer membrane, you now have your LPS inserted into that specific region here, and that sits on the outside and contributes to pathogenicity of these guys. So that's the perfect place to stop. Next time we actually will do a gram scale lab. Let's say